So let's get started. All right, good afternoon, everyone. My name is Huang. I am the postdoctoral visiting fellow from Biostatistics and Bioinformatics branch of NICH at NIH. So it is my great pleasure to be here with you and also to attend such a fruitful workshop. So my today's topic will be focused on a differential abundance and correlation analysis of microbiome data. Specifically, I would like to talk about the challenges of performing such analysis and some solutions we provided so far. So first of all, we know like human health is a very complex issue. So basically you will be affected by several factors, either external factors such as the diet, physical activities, or genetic features, or the internal environment, such as the microbiome. And these factors interact with each other and accumulated by time all contribute a very significant part of our human health. But as you all know, today's topic will be focused on the microbiome part. So let's zoom in to the microbiome. When you talk about microbiome cells, like we all know, it's more than 10 times of microbial cells than human cells. And if we talk about genes, we only have 20,000 human genes as compared to 2 to 20 million microbial genes. So indeed, we have an elephant in our gut. So before we jump into the details of statistical analysis, so I would like to briefly discuss about how the microbiome data being generated, and that will help us understand what is the parameter of interest we'll be focused on. So first of all, we have an ecosystem of interest, such as the gut. And based on this ecosystem of interest, we can draw a random sample out of it, such as a fecal sample. Then we can do such as the 16S RNA sequencing or shotgun sequencing and do a library preparation. And at the end of the day, it will have something like the read count for so-called operational taxonomic unit OTU or applicant sequencing variant ASV. And these OTUs or ASVs can be further summarized at different phylogenetic levels based on our research interest, such as species level, family level, and so on. So the data will be summarized as an abundance table. So each row will stand for each taxa, and each column stands for samples. So if we get the sample total by each column, which is the sequencing depth or library size, will play a very important role in the following the differential abundance analysis. We will talk about this library size issue more in detail in later slides. Okay, so now we got the data. Now, because of some unique features of the microbiome data, it brings a lot of challenges of analyzing it, especially for the differential abundance analysis or the correlation analysis. So let's take into the details of it. So the first challenge stems from the compositionality, which means the observed data are actually relative quantities than absolute quantities. This is because the high throughput sequencing, HTS, can only deliver reads up to the capacity of its instrument. So basically, if you sum all the observed abundances, it will be equal to the fixed constant, or it's the fixed constant, which is the capacity of the instrument. So this compositionality issue will be very phenomenon if we transform the observed abundances to relative abundances, or we transform into proportions. So here is a toy example. On the left-hand side, we have abundances for 5 taxa. And as you can tell, only the green one are differentially abundant between these two ecosystems. However, if we transform all these observed abundances to relative abundances or proportions, this single change of the abundance will lead to everything change with regard to proportions. So this is the compositional effect of the data. And the second, or the most important feature of the microbiome data is that what we observed are affected by differential sampling fractions. So here is another toy example. Suppose we have 2 subjects, subject A and B. And if we look at their ecosystems, such as what's happening in their guts, however, we have 4 blue and 4 reds in ecosystem A, and we have 12 blue and 4 red in ecosystem B. So obviously, only the blue taxa are differentially abundant or more abundant in ecosystem B. However, if we look at the samples, which is actually the data we are handling, we have 2 blue and 2 red in sample A, and we also have 3 blue and 1 red in sample B. So what is the tricky part is that if we look at the library size in a sample or the sequencing depth in a sample, both sample A and sample B has equal library size. So if we only normalize the data or we account for the data by their library size, we will regard these two samples as well normalized. And then it will lead to the conclusion that both blue and red are differential abundance. But as we know here from this talk example, only normalized data relying on the library size will lead to the false positive. So we cannot only rely on normalization based on library size itself, we also need to take into account the ecosystem microbial load there. So a concept to combine these two ecosystem microbial load and the library size in a sample is what we call the sampling fraction here, which can be empirically estimated by the library size, the ratio of library size and the ecosystem microbial load. And as you can see here, the sampling fraction for subject A is 1/2, and the sampling fraction for subject B is 1/4. So the more proper way to normalize data is normalizing it by the sampling fraction rather than library size. And in our, so now we know there are some unique features of the microbiome data, which leads to some challenges to do the downstream analysis. Now I would like to talk about the first section of my today's topic, which is the differential abundance analysis. And the problem setup will look like that. So first of all, we have a population of people, of course, and then we can draw some random subject out of this population. And we have a unit volume of biospecimen and do some processing such as the sequencing and library preparation. We have our data, which is the observed abundance in a random sample, which we denote as O. So the O is the random variable we can observe. However, what we are interested in is an unobservable variable, we denote it as A, which stands for the true abundance of taxa in unit volume in the ecosystem, such as the gut or oral environment. So how can we link these two random variables together, like link the observable O to unobservable A? So here is the formula we proposed. So basically, given C and S, where C is a taxon-specific sequencing efficiency, and S is the sample-specific sampling fraction. So given C and S, and given the unobservable quantity A, on average, the random variable O will equals to A times a multiplicative factor, which is the product of C and S. And then we can make an inference out of the formula. So the parameter of interest is actually the average of A, which is the true average abundance of a taxa in a unit volume of ecosystem at population level. So we denote it as theta. So both O and A are random variables, and our primary interest is actually theta here in this differential abundance setting. And you can see the C here is taxon specific, right? So if we are interested in differential abundance analysis by each taxa, the thing that will lead to the bias, or the parameter will lead to the bias, is actually S here. So we need to take care of the S very carefully. So in our review paper, which published later in last year in MPG Biofilm and Microbiome, we have summarizing different popular scaling methods to deal with sampling fraction S. So such as the D-SIG2, HR, which are very popular in doing the RNA-seq analysis, they have their embedded normalizing methods such as the median, the upper quantile, or TMM methods. And also there's another 2R package, including metagenome-seq using the CSS normalizing methods, or another standalone R package called the range to do the normalization. However, these methods has an implicit assumption, which is a large proportion of features, such as genes or taxa, are not differentially expressed. This is a very reasonable assumption for gene expression data, because we know that a large proportion of genes are actually not differentially expressed, but it may not be valid for microbiome data. For example, if we aggregate data to some higher taxonomic levels, such as the phylum level or family level, this might not be true for these normalization methods. So because of some limitations of the current existing methods, we propose a new model, which called the analysis of microbiomes of analysis of compositions of microbiomes with bias correction, in short, ANCOM-BC model earlier last year, which is published in Nature Communication. And here, due to the time limitation, I only show you the most straightforward formula for the two-group comparison problem. So basically, the statistical formula for two-group comparison stands for a multiplexed model, which is listed here. So assume the observed abundance equals to the product of sequencing efficiency, C, and times the sampling fraction, S, and times the parameter of interest, theta, and some random error. And we have a least rigorous assumption as all existing methods will only require some taxa are non-differential abundance, not a large proportion of them. And then we do the log transformation to transform the multiplexed model to the additive model. And now we have in log scale, the observed abundance O equals to the sequencing efficiency plus sampling fraction plus the parameter mu and some random error. As you can see here, we explicitly state the sampling fraction effect in our model so that we can not only correct the bias for the parameter of interest, such as theta and mu, but we also get the estimate for the sampling fraction simultaneously. And the computing details will involve in two steps, like algorithms. The first step is iterative least square estimation, and the second step is fitting a Gaussian mixture model and using the EM algorithm to get the estimate. So for the computing details, you can refer to our methodology section in the paper. So here are some simulation results, basically comparing our methods and some existing methods. So here, the first picture shows the normalization efficiency. So basically the y-axis is the residual between the true sampling fraction, which we simulated, and the estimate sampling fraction for the methods. So actually in this plot, we have generated two groups, which denoted by a circle for group 1 and the triangle for group 2. So if the method works well, which means if the method can remove the bias of the sampling fraction, we will expect the circle and triangle will intermix randomly and have a very small range or a small variation across y equals to 0 line. So as you can see here, only our methods, the ANCOM-BC, outperform all other methods, like with the smallest range, the smallest variation, and all the triangle and circle are intermixed with each other. And this is not the case for most of the methods. For example, if we look at the far right box, so this method stands for the total sum scaling, which means we transform the abundance table to the proportions and do the analysis accordingly. And you can see here, this method has the largest variation among all other methods, and the triangle and the circle are well separated with each other. And this is not what we want in doing the differential abundance analysis. So just never transform your data to proportions. And the control of normalization efficiency leads to the FDR control and power maintenance. So we're also using the same simulation settings, we compare different methods with regard to FDR and power. So again, we see here only our methods and the previous version ANCOM can control the FDR well below the nominal level, while have a competing power as compared to other methods. So besides from the simulation studies, I would like to also illustrate the application of ANCOM-BC to a real data set, which is the to review the role of microbiome and some microbial byproducts, such as the short chain fatty acid in HIV patients. And this is the paper. This paper has been accepted to microbiome and will be appear soon. So basically, the data set contains 265 male subjects and ages ranging from 19 to 80. And the subjects were collected from 4 different locations in the US, Baltimore, Chicago, Pittsburgh, and LA. At visit 1, no subject are seroconverted or gets HIV infected. But at visit 2, 109 men were seroconverted. We make it as a C group. And the remaining 156 people didn't get seroconverted, and these will serve as negative controls, the NC group. And if we look at the CD4-CD8 ratio of SC and NC group at both visit 1 and 2, we will see a clear pattern of this CD4-CD8 ratio. So basically, at visit 1, there's no difference of CD4-CD8 ratio, which is a well-known biomarker for inflammation. But in visit 2, we can see there's a statistically significant drop of CD4-CD8 ratio. And this is well expected because these subjects get infected at visit 2. However, this means we can only see the change at visit 2, and we are not able to see the change at visit 1, which means this biomarker cannot be served as a predictive biomarker because at visit 1, CD4-CD8 ratio tells you nothing. The same thing happens if we calculate the alpha diversity. So for example, here we calculate the Shannon diversity index at visit 1 and visit 2 respectively. So at visit 1, there's no difference, but at visit 2, we see a drop in Shannon index among the SC subjects. So again, the alpha diversity cannot be served as a good biomarker for indicative of disease until the subject indeed developed the disease or get the infection. So as a summary, at first visits, when no one is seroconverted, there's no difference in either the CD4-CD8 ratio or the microbial alpha diversity. But we can see a significant reduction in SC group as compared to NC group at second visits for these two biomarkers. So we would like to take a step further. So although there are no difference between SC and NC during the first visit, were these male subjects who later got seroconverted develop immune deficiency before the seroconversion? The answer is yes, because as we check the cytokines, which are the product of immune response, we indeed see a lot of change of cytokines at visit 1, which means something indeed happened before they got seroconverted or at visit 2. However, the cytokine is the product of immune response. We would like to know the biology, like before the immune response, exactly at the immune response, the time point. So here is the question we raised. Because it is well known that microbiota are involved in the inflammation and immune response. So we assume or we propose a hypothesis that men who get seroconverted later on were they predisposed to seroconversion because they have a different gut microbiome composition. So we used ANCOM-BC to compare the SC group versus NC group at visit 1. And we indeed see a lot of changes for these species we identify. So for example, we see an increase of pro-inflammatory bacteria, for example here, from this species from the genus Prevotellaceae, Seroconversion. These are a well-known species found in various gut inflammatory conditions. So we see these species, these pro-inflammatory bacteria increase at visit 1 among SC subjects. Additionally, we also see a lot of protective bacteria decreasing for SC subject at visit 1. For example, as I highlighted here, we have several species which belongs to Bacteroidaceae have been decreasing in the SC subject. And these species of Bacteroidaceae has anti-inflammatory function by promoting T regulatory cell reactivity in a lot of literature. So these results indicating that a pro-inflammatory condition in the GI tract in SC has been like showing even before these patients acquiring HIV infection. So we can also like visualize this table by waterfall plots. So basically these waterfall plots are ordered by their effect size, and ANCOM-BC can also help construct the confidence intervals and significant level for these findings. And except from, in addition to the individual like differential abundance analysis for each taxa, we also know that Prevotellaceae and Bacteroidaceae are a well-known predictor of the inflammation in a lot of literature. So in addition to the individual testing, we also check the difference of this ratio between the negative controls and seroconverters. And if you look at the left-hand side, we indeed see a statistically significant jump of this ratio in the SC group at basic 1. And if we fit the logistic regression model with y equals the probability of seroconversion, we see as the increase of the Prevotellaceae and Bacteroidaceae ratio, the higher chance of this subject being exposed to seroconversion. So which is in accordance to the existing findings in the literature. So except from the microbiota itself, we also know the short-chain fatty acids are the primary fermentation product of gut microbiota from dietary fibers. And this is a major factor in maintenance of gut and homeostasis. So in addition to check the microbiome composition, we also take a look at these short-chain fatty acids. And there are several important short-chain fatty acids, for example, like acetic acid, which regulates the gut pH controls, and the propionic acid, which can regulate appetite and have some anti-inflammatory functions. So if we look at the correlation between a well-known inflammatory indicator, which is the CD4-CD8 ratio versus list of short-chain fatty acids, we can see a very interesting positive correlation here. So the propionics level are statistically significant, positively correlated with the CD4-CD8 ratio. So if you can recall, the higher the CD4-CD8 ratio means these subjects are more likely to be in the negative control group. And the higher level of propionic level also stands for a more strong anti-inflammatory function. So that's indeed a very interesting biology behind the short-chain fatty acids. So this is what we propose out of these real data sets. So here is a potential hypothesis we proposed. So basically, the chance of getting infected relies on subject whether they have a poor, quote, poor gut microbial composition and short-chain fatty acid products. And if so, this will lead to a reduced immune response, such as the CD4-CD8 ratio and cytokines respectively. And these patients will be exposed to seroconversion with higher chance, and at the end of the day, it will lead to a disease with a higher chance of acquiring the disease. So this is the potential hypothesis we form based on what we find. And the last section of my today's talk is the correlation analysis for microbiome data. So why are we interested in doing the correlation analysis? This is because in many microbiome studies, the primary goal is not the differential abundance, but to discover whether there are interactions among microbiota within or between ecosystems, such as the gut or oral environment. And the correlation analysis is, of course, an important starting point. However, the estimation of correlations among microbiota is also a challenging problem due to the unique features of microbiome data, as I illustrated earlier, such as the compositionality, the unequal sampling fractions, and also, of course, a well-known like excess zeros. The microbiome data is a typical sparse data. And it's reasonable to assume most taxa are uncorrelated, especially in some like lower taxonomic levels, such as the species level, which means the correlation matrix should be a sparse matrix once we get the results. And lastly, not all interactions among microbiome are expected to be linear. They can have some nonlinear relationships. However, the existing methods such as the standard Pearson-Spearman correlations or a well-known methods proposed in 2012, which called sparse-CC, to analyze the correlations among microbiome, they are all focusing on detecting the linear relationships. But we cannot expect that all the relationships are linear. So it may have some nonlinear relationship existing there. So because of that, we would like because of these limitations of existing methods, we would like to propose a more comprehensive tool of analyzing the microbiome correlations. So here is a illustrative example. So suppose we have two taxa, like taxa called T3 and taxa T4. And this is, on the left-hand side, is the underlying true relationship between these two taxa. As you can see here, these two taxa are actually not correlated at all, with the Pearson correlation almost equals to 0, because I simulated that way. So, and the data set was simulated from the negative binomial distribution. However, if we draw these abundances from the ecosystem to what we observe, what the data actually is, like in the observed abundance data, as you can see, we have a very strong positive correlation between these two taxa. And this strong correlation is not due to the underlying biology, which is here. The underlying biology means they have no correlation, but it's due to the library size effect, or the unequal sampling fraction effect, or the compositional effect. So here we have to take care of these effects due to microbiome data and recover the true correlation information here. So here is a simulation analysis. Basically, we simulate 10 taxa, and the taxon 1 and taxon 3 are perfectly linearly correlated, and taxon 2 and taxon 4 are perfectly quadratically correlated, and all the remaining pairs are uncorrelated, as shown here in the left-hand side. Again, the data was simulated from the negative binomial distribution, so you can see we have some long tails in the microbiome abundances. And from these true abundances, we can draw a random sample out of it to construct the abundance table. And based on this abundance table, we calculate the Spearman correlation. So if we don't do any adjustments and calculate the Spearman correlation using the observed data, this is what we got. So the block coded in green means true positive. So we indeed detect the linear relationship between T1 and T3 here. So we have a green code for this one, which is the true positive. It also has a false negative. The Spearman correlation is not designed for detecting the nonlinear correlation. So not surprisingly, it fails in detecting this quadratic relationship between T2 and T4. But what's even worse is that it has a lot of false positives. If you remember in our last example, the T3 and T4, these two guys are actually not correlated. But if we draw it to the sample, because of the bias introduced by unequal sampling fractions, these tend to be positive correlated. So which leads to the result here. We have a lot of false positive resulting from the unadjusted Spearman correlations. And of course, the remaining gray blocks are true negative. And we apply the same data to SPAR-CC, which proposed in 2012, specifically to detect the linear relationship among microbiome data. So as you can see, the SPAR-CC managed to detect the true linear relationship between T1 and T3. So it has a true positive color here, the green color. However, because of its design, the underlying methodology, it failed to detect the quadratic relationship as well. So it has ended up with a false negative block here. But the SPAR-CC has much better performance in controlling the false positive as compared to applying the Spearman correlation directly. So we know that neither SPAR-CC nor the Spearman or Pearson correlations can identify the nonlinear relationships. So what we can do is to borrow the idea from what we call the distance correlation, which proposed in 2007. So the limitation of Pearson correlation and Spearman can only test for linearity in relationship. While the distance correlation proposed by Zuki in 2007 allows us to test for nonlinear relationship in general. So our methods will generalize the concept of distance correlation for microbiome data, while also account for the features of the microbiome data to detect both linear and nonlinear relationships. And the model is very similar to a model setup is very similar to ANCOM-BC. So I will skip the details here. So let's show you the results of our methods, which called SECOM, the sparse estimation of compositions of microbiome. So not surprisingly, our methods, because we use the distant correlation concepts, it can not only detect the linear relationship, but also the nonlinear relationships between microbes. And also it has a well control of false positive, and remaining pairs are recovered as true negatives. So finally, I would like to illustrate the usage of CCOM using a well-known real data, which is the global gut microbiota data. So this data consists of more than 10,000 OTUs obtained from subjects from three different locations: the US, Malawi, and Venezuela. And because the subject from these data sets has a wide range of ages, we only subset the age less than 2 years old as the infant babies. So, so the resulting data set consists of 50 babies from the US, 47 babies from Malawi, and 27 babies from Venezuela. And the OTU data were aggregated into species level for analysis, and the top 20 were selected for visualization. So here is the results we have for the correlation we estimate we got. So the color coding here, if the green color means the pair are not correlated, and the green color, the red color means these pairs are positively correlated. And if we see the blue color, such as here, the more dark blue color, it means these pairs are negatively correlated. So as you can see, for these babies, for infants, more pairs of species appear to be positively correlated. And if you look at this cluster here, these are several species belongs to the genera Bifidobacterium that is commonly available in breast milk and show a very strong positive correlation with each other. So these species belongs to Bifidobacterium form a kind of like a potential cluster with each other. And this potential and this cluster may represent a mutualism or a commensalism for this species. Again, we see a similar a cluster here, like these three species are strongly positively correlated with each other and also form groups in addition to this group with Bifidobacterium species. So as a summary, analyzing the microbiome data is a very challenging problem due to their unique features, such as the compositionality, differential sampling fractions, and of course, the sparsity with zeros. And both ANCOM-BC and ANCOM can correct the bias due to these features and lead to an unbiased differential abundance analysis or the correlation analysis, respectively. And what is important to emphasize is that ANCOM-BC and CCOM are designed for drawing inference on the absolute abundances, not the relative abundances, such as the proportions. So, and last but not least, CCOM can not only identify linear relationships, are also able to detect nonlinear relationships among microbiota, which fills an important gap in the literature. And I would like to thank my supervisor, Dr. Shriver, with his tremendous help and guidance in doing this methodological work, and my collaborator, Dr. Charles Renato and Dr. Yu Chen from University of Pittsburgh. And that is all I have for today's presentation, and I'm happy to take any questions.